Place an ice cube in a glass of water, and it floats. But in this glass, the ice sinks directly to the bottom. The clear liquid causing this anomaly is heavy water, or D2O. It is visually indistinguishable from standard light water, H2O, but its physical properties diverge immediately. The difference lies in deuterium. Deuterium is a stable isotope of hydrogen that contains one proton and one added neutron. This extra mass makes the resulting liquid approximately 10% denser than standard water. Chemically, deuterium is identical to standard hydrogen, also known as protium. It engages in the exact same metabolic reactions and forms the exact same molecular compounds. Yet animals cannot survive drinking only heavy water. Sustained consumption of D2O over the course of 2 weeks will reliably kill a mammal. The addition of a single, uncharged subatomic particle alters the physical reality and biological viability of the entire solvent. In complex biological systems, heavy water exhibits extreme cytotoxicity. Replacing just 25% of the protium in a mammal's body water with deuterium halts reproduction, inducing total sterility. Pushing the concentration above that threshold triggers rapid organ failure and lethality within a matter of weeks. At the cellular level, D2O specifically targets actively dividing cells and physically arrests the mitotic period. Mitosis is the phase of the cell cycle where replicated chromosomes are separated into 2 new nuclei. We can trace a direct line of causality for this biological failure. It begins with the quantum mechanics of a single neutron, scales through physical chemistry, and ends with the mechanical halting of the cell cycle. The water inside a cell operates as an active physical structure dictating the boundaries of life, rather than a passive backdrop for chemical reactions. The mechanism begins with the quantum mechanical well and the kinetic isotope effect. A quantum potential well is a mathematical region surrounding an atom where its energy is lowest, determining how tightly it is bound to its neighbors. The kinetic isotope effect refers to the change in a chemical reaction's rate when an atom is replaced by its heavier isotope. Because the deuteron has twice the mass of a proton, it sits deeper within this energy well. A standard proton's higher position makes it easily perturbed. When a neighboring water molecule approaches, it is drawn away, whereas the shielded deuteron remains highly resistant to perturbation. This creates our first structural data point. Light water has an intramolecular bond that is 3% longer than the corresponding bond in heavy water. Second, the intermolecular hydrogen bond connecting 2 separate molecules is 4% shorter in light water. These combined variances warp the geometry of the molecules. Light water forms a broader, highly asymmetric structure. Heavy water assumes a narrower, highly constrained tetrahedral shape. These minute structural differences were imperceptible for decades, only recently verified using advanced X-ray and neutron diffraction techniques. The added neutron creates a tighter, more rigid geometry at the subatomic level that cascades upward into macromolecular physics. This geometric rigidity directly impacts the mathematics of chemical reaction rates. In 1947, chemists Harold Urey and Jacob Biegeleisen independently modeled how isotopic mass shifts equilibrium fractionation. The Urey-Biegeleisen-Mayer model provides the mathematical proof. It relies on the reduced partition function ratio, a variable expressing equilibrium partitioning behavior based on vibrational frequencies. The model proves these functions are unequal. A heavier isotopic mass lowers the frequency of molecular vibration. In a physical bond, deuterons form stronger covalent attachments that require significantly more energy to break than standard hydrogen bonds. By requiring more energy to initiate reactions, heavy isotopes lower the thermodynamic speed limit of biochemical processes. Moving from mathematical models to living cells, this physical chemistry alters the hydration shell surrounding all proteins. A hydration shell is a continuous, dynamic layer of water molecules that completely encloses and interacts with a dissolved protein. A protein's ability to fold into its functional shape and perform mechanical work relies entirely on the flexibility of this surrounding water layer. When D2O replaces H2O, the environment of the hydration shell immediately hardens. The highly rigid tetrahedral structure of heavy water drastically alters the thermokinetic parameters of the macromolecule, physically restricting its natural movement. This rigidity severely limits conformational freedom, slowing down the vital proton exchange required in the active centers of enzymes. Heavy water places the cell's mechanical machinery inside a thermodynamic straitjacket, making biological movement prohibitively costly. The consequences of this thermodynamic straitjacket are most lethal to the single biological structure most dependent on rapid, flexible physical assembly, the mitotic spindle. During cell division, a protein called tubulin must rapidly polymerize to build microtubules. These microtubules act as cables to physically pull chromosomes apart. This micrograph displays sea urchin eggs developing in deuterated environments, allowing researchers to observe the exact point of failure. Initially, researchers assumed the spindle was deteriorating or melting under the metabolic stress of heavy water. The observations revealed a counterintuitive anomaly. D2O accelerates tubulin polymerization and hyperstabilizes the microtubules. This triggers a cascade of errors. During prometaphase, the stage where the nuclear envelope breaks down, this extreme stability delays the necessary alignment of chromosomes on the equatorial plane. During anaphase, the stage where chromosomes are pulled to opposite poles, the hyperstable spindle fails to retract properly, elongating abnormally instead. Heavy water stops cell division. It does not break the mitotic spindle, but rather freezes it in a state of hyperstable perfection. Modern research indicates that eukaryotic cells act as highly sensitive deuterium to protium gradient sensors. All biological life is evolutionarily tuned to the natural environmental baseline ratio of exactly 150 parts per million of deuterium in surface water. Moving up the gradient exposes cells to deuterium-enriched water, or DEW. As demonstrated, DEW mechanically arrests cell division in the G2M phase due to tubulin hyperstabilization. The G2M phase is the final checkpoint before a cell divides its nucleus. Exploring the inverse phenomenon means moving down the isotopic gradient using deuterium-depleted water, or DDW. Neoplastic or tumor cells rely on altered metabolic pathways. These pathways produce anomalous amounts of intracellular water as a byproduct of rapid energy consumption. Introducing DDW disrupts this specific mitochondrial water production in tumors, triggering severe oxidative stress inside the malignant cells. Shifting the isotopic baseline away from 150 parts per million acts as a targeted structural weapon, triggering immediate functional crises in highly specific cell types. The DDW-induced oxidative stress inside the tumor cell dictates a downstream genetic consequence. This stress forces the rapid downregulation of critical anti-apoptotic survival genes, such as BCL2 and MYC. BCL2 and MYC are specific genes responsible for producing proteins that block cell death and promote unchecked growth. This diagram illustrates the divergent endpoints. The genetic collapse on the bottom path forces the tumor cell into G1 phase arrest. The G1 phase is the first resting and growth state of the cell cycle. Stripped of its survival genes, the cell undergoes programmed death. A single extra neutron deepens a quantum well. That shift restricts molecular geometry, imposing rigidity on the hydration shell, which mechanically halts the cell cycle. The subatomic mass of a solvent's hydrogen isotopes actively dictates the physical boundaries of cell division. In biology, physics is destiny.